Hello everyone, welcome to Data Visualization Chapter 7. This is the Chapter 7 of Python for Biological Data Analysis. My name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. So let's start how we can perform different types of visualization on biological data sets. So first of all, you need to open your Chrome and just simply type Google Colab. You will have this kind of window, like here. Now, like GitHub, click write ASH 100, press enter. Click this part, scroll down, Python for Life Sciences. Scroll down to Chapter 7, Data Visualization. Click here. Okay, now click this connect button. So, learn Python for biological science. My name is Dr. Ashfaq Ahmed, and during teaching, I will use examples from the field of biology or life sciences. Therefore, if you are a student of computer science or any irrelevant field, I would highly advise this course is not for you because I'm trying to use examples from biology with some basic material or with some basic coding. So it could be boring for you. Our course contents are these are the chapter-wise contents we have covered so far. Chapter 1, getting started, are a foundation chapter, control structure, data structures for biology, working with files, NumPy, and pandas, and data visualization. Today we will complete the data visualization part, and then we will proceed to some biological applications and some practical chapters. Okay, so what actually is data visualization and what is Matplotlib? Matplot is the name of a program and lib stands for the library. So Matplotlib is a Matplot library. Okay. So far, I assume that you will be somehow aware of the term library because before this we had used NumPy library and we had used pandas libraries and their functions. So today we will explore in particular Matplotlib because it is very friendly with Python and it is very easy to use or to understand. Okay. So in biology, visualization or visualizing your data is very much important because normally the data in biology is a kind of huge data, or I would call it messy data. So to extract meaningful information from a huge data or from a messy data to make some hypothesis, to comment on the data, you need some meaningful visualization or some meaningful visualization techniques. Apart from the visualization, you also need to produce a high quality visualization because as far as I know, the scientific journals publishing biological or biological relevant data, they need the minimum requirement of 300 dpi, which is still on the higher side. So Matplotlib will produce the higher dpi as much your machine or your computer supports. In particular, today we will discuss about the line plot, histogram, scattered plot, and box plot. Why visualization matters in biology? I already discussed this briefly. Okay, so getting started with matplot or matplot visualization. In our last video, we import pandas as PD, and in our chapter 5 video lecture, we imported NumPy. Today, we are increasing another import, that is import matplotlib.pyplot as a PLT. Okay, so when we run these three statements or these three codes, all these three library will be imported in our workspace and we will be able to use them. And optional set as a clean style or some wide grid, so that's okay. If you ignore it, also okay. Otherwise, just run it. Okay. Now you can see all these three libraries are imported into the workspace. So now you need to understand this. Uh, I'm sorry. Here, I'm going to generate a synthetic biological dataset. That means I'm generating a virtual kind of dataset which do not exist, but for this tutorial, I will generate it. Also, when you deal with your experiment, most of the time or almost every time, you must have your own data. Then you will not need to generate a synthetic biological dataset. You will just upload that dataset, and that is very easy. Okay, so therefore, I will try to explain this data command, like how I'm doing or what I'm doing. I'm using random seed 42. And what is NP random seed 42? I will tell you later. I'm trying to generate a list of 100 genes with two different treatments. To the one I call it treatment A, to the other I call it treatment B, and okay. And the third one is control. Control is actually the baseline. Okay. So let me explain these codes one by one for you. We are simulating expression level for 100 genes. Control will be baseline expressions, and treatment A will be slightly increased expression, or you can say upregulated expression, or the treatment B will be slightly decreased expression or down-regulated expression. This dataset I wanted just to mimic how genes responds under different conditions or under different treatments in your experiment. Okay, so I use np.random.seed42 that actually set random seed, that every time any one of you when runs this notebook, they will get the same random number, so it will ensure the reproducibility. Okay, this line actually explains like we are creating the list of 100 genes from gene 1 to gene 100. And here, this line in the code, it simulates the expression level of 100 genes under a control condition. Okay, what does it mean, the control condition? If you see, I use LOC. The LOC or log means the average expression is 50. So I asked the program to keep the average expression 50, just like for some give to 38, to some 32, to some 56, to some 59, but keep the average around 50. Okay. And the scale 10 means that the standard deviation, keep the standard deviation up to 10 in the values. Then for the treatment A, I use control plus this statement. That means that the treatment A, by adding a small increase to the control value, increase of 5 number, like if For a gene, if a gene in a control is let's say 48, the expression is 48, then that gene will be 48 plus 5, 53 in treatment A. And again, in treatment B, as I mentioned, that I'm trying to create a list with the down regulation. So the LOC I used LOC minus 5. That means if for a gene, if its expression in base line or the control, if its expression is minus 48, in treatment B, it will be 43. Okay, 5 reduced. And then finally here, if you see, I group the genes, control, treatment, and treatment, these three conditions differently as a single DF or as a single data frame. Okay. Let me remind you again that in your case, you will not need to generate a hypothetical data set like I'm generating. You will use your own data sets. So why I'm not using my own data set? Because I'm not sure this course will be taken by a person from molecular biology or from bioinformatics or from microbiology or from plant sciences, so that will reduce their interest level. So that's why I just generate a random data set with kind of related to more related to biotechnology. But that's okay if you are a plant biologist and you have some data set of plant sizes, plant growths, or something else, the nourishment value, that's okay. Only the names will be different in the data set. There will also be some values. Okay. Now we are going to draw the plot for a single. Okay, I forgot to run this. Okay. Now the data set is generated and top five are the starting five genes were how to say genes were mentioned here by the df hit function. So you can see gene 1 control is this is the expression, treatment A, this is the another the treatment, and then the treatment B, this is almost the same. Okay, but we don't care, this is a data set. Okay, now uh, I'm generating a line plot for a single gene, and here I mention a gene index 10. So let's say run it. Now, it will give you a fair idea if you are interested in a single entity in your dataset or in a single gene in your dataset. So you can see for gene expression of gene 11, it will be, you can see it's a control here, and the treatment with treatment A, its expression goes down, and in treatment B, its expression goes up. Okay. Now, if you are interested in some another gene, if you are confused that why it says gene 11, so that's okay. Wait, let me tell you. Gene index 12. Okay. Now for the gene index 12, you can see the control is here, the treatment A is up and the treatment B is down. But here it says the expression level gene 13. So the point is that in Python, the dataset normally starts from 0. So the gene 12 index is the gene 13 in the dataset. So that's the reason. So you can play with it. Let's say I'm interested in gene index 89. It will be gene 90 here. So the gene index 89 is the control. It's here. Treatment A is here and treatment B is here. So how well you can simulate or you can find out the relationship between different treatments are different variables. In fact, I will use the word variable. So, so easily you can find out the relation between different variables for a single entity, because in our data set I told you that I have used 100 genes. So this data set contain 100 genes, although this is a hypothetical data set. Okay, now let's try this for all of the genes, not a single gene. So let's say we have a control. In our data set, we have three groups: control, treatments, and treatment B. So now I'm trying to explore a box plot for all the genes. Now you can see, if you see this orange line between the box, this is a median. And if you see these tails, the down and up, these are normally the standard deviation. So if you see in the condition, the median is just below 50. In treatment A, it's above 50, and in treatment B, it's much more below 50. It was possible because during the dataset generation, I told you, like, I'm creating the treatment B with some down-regulated genes. So of course, in treatment B, the expression level will be downward. Okay, so this is for the median of the overall expression of these 100 genes in the dataset. Okay, now let's try a histogram and let's see what we see. So this is the histogram expression level, and normally we use the histogram expression or the histogram chart when we are interested in a kind of distribution. If we want to show some distribution in the data, for example, in our whole data, the maximum, the maximum, like if you say the frequency, 14 genes, or I would say 15 genes. 15 genes recorded the expression of, if it is, I consider it 52. So 52 expression was the most or the highest, not by expression, the highest by number. So the distribution majority was 15 genes. They recorded almost the expression of 52. Then 13 genes, they recorded almost 54 expression, and there was expression up to 80, but only one gene. You can see here the frequency is down. Okay. Now, if you see this code, I just plot dot histogram, and here I just mention plot dot box plot. These are the functions of the Matplotlib library. Matplotlib, and it's very easy. Just plot dot histogram, then data frame, and in the data frame, we ask to control treatment A. So this was the chart only for treatment A. What this bends mean? Ben actually divides the data into interval. Let's say you want 10 bends. So now you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 10 bends. Then now the expression level, you can see it 25 because the bends are reduced. So now from 50 to 55, there are almost 22, 27 genes or 28 genes. So the same way you can reduce. Let's say I'm interested in a more like 30 bends. So you can see now these are the 30 bends. Let's see, I'm interested in the 40 bends. Now you can see 40 bends, and to be exact, the frequency of 12 genes up to 52. Okay, so also if you increase the bends number, then you can also see the missing columns, because if you consider this a 78, so we did not have a 78 expression for any gene. So this was about the bin. Now, if you are interested to change this color, so let's say it's light green, you can change it to red. Let's see what's going on. Now you can see. Uh, Another thing, okay, these are actually the titles. Now, if you see, I used a treatment A. Now let me change it to treatment B, and this is the treatment B with 40 bins. Now I'm interested to change to see the control, but if you see still this legend or caption is treatment A. So you will, as we are using the control group, so you can just change it to the control. Now you can see distribution of expression in control group. So that is how you will manipulate these smaller things and to extract a meaningful figure with colors, with bins, with sizes, each and everything. And these are the labels. Okay. Now I'm interested to plot a scatter plot comparing two conditions. So in our data, we have three conditions: control, treatment A, and treatment B. So now here I am running this thing. So now expression comparison control versus treatment A. So if you see the control and treatment A, each dot represent a single gene. So how you will interpret it? For example, this is we are going to interpret this dot, this gene. So in the control, its expression is almost 45. You can see, but in treatment A, its expression is almost 41. Okay. Now, if you are interested to change it to treatment A to treatment B, now you can see plot changes because now we are dealing with control and treatment B. Because I did not change it to B, so still now you will see it. It's a treatment B with control. If you are interested between treatment B and treatment A, treatment, now let's see. This is the treatment A and B. Why you are seeing still a control? Because I did not change it here, and you need to just treatment A. Now you can see, and this what it does. Let me show you. It increase or decrease this transparency threshold. Okay, this alpha value, and this is the overall title you will use. Okay, so in this tutorial or in this chapter, I showed you four types of different plots on hypothetical data sets of 100 genes with control, treatment A and treatment B. And we explored the line plot. Remember, the line plot we will use normally for a time series data, just like if you are um, observing some kinetics, some growth curves for a plant or for a gene or for whatever in your data set, then line plot turns out the best. If you are using a growth, representing a growth curve and you are using a histogram plot, it will not be so meaningful or so explanatory. So this is also must which plot to use for what kind of data. The histogram plot, normally we use it for the distribution. Like I did this, I explained this for you. The scattered plot, this one is the scattered plot. So normally we use the scatter plot for the correlation between different variables. In our case, we had three variables, control, treatment A, and treatment B. So we use the scatter plot to compare different variables. And finally, the box plot, we used it across the comparison of different groups or different variables, you can say again. So overall, you can see here, this is actually the box plot. So that was all, and I hope you will now be familiar from the Matplot or Matplotlib, and you will also be aware that this is not a rocket science. If you are seeing some eye-catching visualization in different manuscripts or papers, you can generate your own manuscripts, your own visualization or high quality visualization using Matplotlib for your datasets. Thank you so much. See you next time. Bye bye.